Hey guys, welcome back to another 2013 IBO paper question. This one is going to be a little difficult, so I want you to pay close attention. Okay. Now, first of all, let's start with the question. 3 yeast strains of Saccharomyces cerevisiae have been engineered to obtain a gene for a different enzyme from glycolysis, TPI, ENO, and PYK under control of doxycycline repressed promoter. Okay. First of all, I will get started the question from this point. Okay, so here this is the glycolytic pathway. That is the conversion of glucose into pyruvate. So we have certain substrate, fructose 6-phosphate, fructose 1,6-bisphosphate, DHAP, dihydroxyacetone phosphate, now 2 and 3 phosphoglycerate, phosphoenol pyruvate, and pyruvate. You know, each of those 10 steps, only a few of which are listed here, is controlled by enzyme PFK, AI, TPI, ENO, PYK. Now, what has the experimenter done? The experimenter has created 3 variants of yeast, and in each variant he has mutated one enzyme. So in the first variant, he has mutated the TPI enzyme. In the second variant, he has mutated the ENO enzyme. So TPI, ENO, PYK. Okay. So in each variant, only one enzyme has been tweaked, changed. How has the change been done? What will be the effect of the change? The effect of the change is it makes it doxycycline repressible. That means you can add doxycycline, it will do something to the promoter, because of which the enzyme will not be expressed. Okay, and concentrations of some metabolites are measured in each yeast strain grown on glucose at different doxycycline concentration. So each of these tiny graphs that you see, what has happened? In time, we have increased the doxycycline concentration and seen what happens to the concentration of the substrate. So this graph talks about the concentration of F6P, fructose 6-phosphate, when the concentration of doxycycline has been increased. That is why you have so many graphs, right? So you are measuring 1, 2, 3, 4, 5, 6 metabolites as the concentration of doxycycline was increased for each metabolite in each of those strains. Now let's try to read each of these statements and try to understand and answer them. Okay. First, down-regulating any of these enzymes disrupted metabolite concentration in the whole pathway. In the whole pathway. That means all the concentrations have been changed. Now you can clearly see that if we change the TPI enzyme, we are observing the change in one and a few others, right? Not in all over. The same is the case of ENO. The same is the case of PYK. Not the whole enzymatic pathways have been shaped or misshaped. So this is clearly a false statement. Second, down-regulating any of these enzymes affected the concentration of its substrate more than the concentration of the product. So once again, they are talking about all. So let's examine them one by one. So TPI, if I change TPI, DHAP concentration is affected more than the 2+3 PG concentration. So it's true for this. Okay, yes, the reactant is affected more. In case of ENO, this is the one we are talking about. The concentration of the reactant, in this case 2+3 PG, is affected more than the product. So that's true. That's true. In case of PYK, this once again, yes, the reactant has been affected more than the product. So going back to our question, this is a true statement. Okay. These were the easy questions, guys. Now I want you to focus and answer the next ones. The third question is the equilibrium of the reaction 2 plus 3 Pg to PEP is more on the side of the product than the equilibrium of the reaction from FBP, fructose bisphosphate, to DHAP. Now, what is equilibrium? You need to understand the concept of equilibrium. So, substrate gets converted to product. This is a reaction. Now, most of the reactions occur in both the directions, and most enzymes, the catalyst, they catalyze in both the directions. Okay, at one point what happens is the concentration of substrate and the concentration of product become nearby constant. That means they are not changing. The concentration is not changing doesn't mean that the reaction has stopped. It simply means that the rate of forward reaction and the backward reaction has achieved a same rate. That means at the same rate, the substrate is being converted to product and the product is being converted to the substrate. Now, that does not mean that the substrate concentration should be equal to the product concentration. No, it does not mean that. It simply means that the rate of reaction is the same. Now, if the reaction is more favored towards the product side, you will have more product towards the substrate side. When does this happen? When the conversion reduces the delta G. What is delta G? The change in the free energy. Now we are going to use this information to get back to our question. Okay, so the question is asking you the equilibrium of the reaction 2 plus 3B Pg. So they are asking about 2 PEP. So this is the reaction they are talking about right now. Okay, now be a little bit vigilant here. Is more on the side of the product than the equilibrium of the reaction FBP to THAP. FBP to DHAP. Now, let's see what happens to these reactants if we are changing their products. How do we change the concentration of their products? You change the rate at which they are utilized in the next reaction. Okay, so if we block TPI, what will happen? DHAP consumption will reduce, right? Blocking TPI reduces the rate of consumption of DHAP. Similarly, blocking PYK reduces the rate of consumption of PEP. Okay, now this is where we are blocking PYK. Okay, so for this we have to observe this line, and this is where we are blocking the TPI. So for this we have to observe this line. Okay, now what do we have to observe? We have to observe what happens. Okay, so you see when you have blocked the TPI pathway, the product is increasing, but the reactant did not produce a significant distortion. Okay, in the case of FBP to DHAP, when you have knocked out the TPI, it resulted in the increase in DHAP concentration, but it did not produce a distortion in the FBP concentration, which means that the reaction favors, or reaction equilibrium favors, the production of DHAP. Okay, but in case of 2,3-PG to PEP, you see that a distortion in the PEP did produce a distortion in 2 plus 3-PG. Okay, so the equilibrium on the reaction 2 plus 3 to PEP is more on the side of the product? No. FBP to DHAP is more on the side of product. That is why it did not produce any response, right? So equilibrium is in the direction of the product. So this makes the statement false. Okay. Now, if this is clear, the fourth will be easy. The concentration of F6P is expected to be unaffected by a downregulation of the AL. Okay. Fructose 6-phosphate to fructose 1,6-bisphosphate. This particular reaction they are talking about. In this reaction, you are using the hydrolysis of ATP. Okay. This will decrease delta G. That means in this case, the formation of FBP or fructose 1,6-bisphosphate will be favored. Okay. So even if we downregulate AL, and the concentration of FBP will increase, it will not distort F6P because equilibrium favors the formation of FBP. Okay. So yes, it will remain unaffected. True. Okay. I hope this question was a challenge for you because that's how we increase and improve, right? Okay. Guys, do check out the book NCERT Mapped. It's there on Amazon. It will help you ace your preparations really well. Take care. See you next question.